So what I'm coming on to talk about is mechanisms of long COVID, which I did allude to earlier. And as an academic, you'll understand that I find this area particularly interesting. So I've put together, well, it comes in two halves, really. The first half of my talk is going to be about what the studies show on mechanisms. And you remember that I talked a lot about syndromes, and I can assure you there is not a syndrome in sight in this lecture. And the second part is my own view of what I think is most likely to be going on, and a kind of personal journey of my research into it, which has been absolutely riveting for me. You're all practitioners, so I don't expect you to share it, but hopefully I'll inspire you a little bit anyway. So the first thing, we're going to look at the medical hypotheses about the mechanisms, and I've broken it down into four parts. There's the effect of SARS-CoV-2 itself, the direct after effects of it, the indirect effects, and the contributory factors. Now, you won't see this chart anywhere else. I put this together myself. Hopefully it will make some sense. And it seemed to me to be that these are the most likely mechanisms for which there is actually quite good evidence that all of this is going on. So let's have a look, first of all, at the effect of SARS-CoV-2 itself. And we'll start with viral reservoirs, which I think a couple of people have mentioned already. So these are places in the body where virus-infected cells can remain hidden from immune elimination, and particularly within the gut and gut-associated lymphoid tissue. So all the time that the immune system is really working well, these viral reservoirs will remain dormant. But the minute that the immune system is under-functioning for whatever reason, then there can be reactivation of any of these viruses in the viral reservoirs. So let's see if this is happening in SARS-CoV-2. Well, yes, it is. SARS-CoV-2 reservoirs in long COVID patients have been found in multiple organs: the brain, the lung, the myocardium, lymphoid tissue, particularly the gut, and also in the central nervous system. So that's a little worrying. And there have been several reports of relapse or reactivation of SARS-CoV-2. In the early days of the pandemic, we heard a lot about reinfection. Scientists have broadly reconsidered now and realized that it wasn't, in fact, reinfection with exactly the same endogenous virus. What it was reactivation of the virus from a viral reservoir. So all the times early on we saw reinfection, no, it actually probably wasn't, but we have to be careful about that distinction. The next point is SARS-CoV-2 can be a super antigen, and I've never heard of a super antigen before. So I discovered that they have potent stimulatory activity for CD4, CD8, and other T lymphocytes, and they can avoid normal antigen processing. Okay, that sounds a bit serious. So does this actually happen in SARS-CoV-2? Yes, it does. Here's a study which found that it was actually called a super antigen. The virus overstimulates the antiviral immune response and induces negative feedback loops, which allow the virus to persist. And it's actually been found specifically, do you remember I mentioned MIS-C this morning? It's a rather nasty inflammatory condition in children, so it's actually been found in that. The next point is delayed viral clearance due to immune exhaustion or lymphopenia. So persistent viral shedding was associated with a reduced number of adaptive immune cells. So that's always a good clue. And viral persistence was associated with significantly lower levels and slower generation of antibodies. So there's probably going to be some overlap here with the lymphopenia and, for example, SARS-CoV-2 being a super antigen. But nevertheless, there's quite likely to be several of these mechanisms going on at the same time. It doesn't have to be just the one. Persistent viral fragments triggering inflammation. So viral fragments are not a viral reservoir. They are proteins from the virus, normally the spike protein, but not always. And they can persist for months in the body and promote inflammation. They may be in viral reservoirs, but not necessarily at all, although they do seem to be fairly prevalent in the gut. So they're not the viral RNA, and they're in, yes, the gut, kidneys, and lungs, they've been found. And here we are, the spike protein found in long COVID patients up to 15 months post-infection. So these were patients who were not testing positive, but nevertheless, here they were, they had the spike protein 15 months after infection. Antibody-dependent enhancement, we've seen quite a lot about this. So this is where antibodies target a virus, but they don't neutralize it. So if you're not clear on how this works, if you generate neutralizing antibodies, they will neutralize the virus, and the virus is, well, I mean, it does what it says, really, neutralize, so that's fine. But the problem is when the antibodies are non-neutralizing, then what they do is they actually facilitate the virus. So they take it into the cell, process called endocytosis, they enhance viral replication, they activate the immune system, and it's a cytokine storm, basically. So that just goes mad, antibody-dependent enhancement. Right, now we'll look at the direct after effects of SARS-CoV-2. And the first and obvious one is tissue damage. So we know that the virus in acute COVID does a lot of damage to the organs. We saw that this morning. And these are not going to recover in a hurry. As we saw, a lot of the problems were still persisting 6 months to a year later. So we're talking about heart, kidney, lung, liver, almost anywhere really. Persistent excess inflammation. Now, I've mentioned inflammation a lot today. So how do we get the inflammation? Well, it can result from two possibilities, really: an overactive immune system or an exhausted immune system, lymphopenia or lymphocytopenia. So an overactive immune system, we've just seen that with antibody-dependent enhancement and super antigens, but let's look at the lymphopenia. So this causes immune activation and release of excess inflammatory cytokines, and it's a compensatory response. Because the T cells are not doing what they should be doing in neutralizing, not neutralizing the virus, but sending out killer cells, instead, they're sending out cytokines. So it's basically the immune system trying to do its best, but it's sending out, it's almost making things worse by sending out things that are just not helping. So that's how it can both be true: overactive immune system and an underactive immune system. Then we've got overactivation of the renin-angiotensin system. So we know the virus binds to the ACE2 receptor. This leads to a reduction in the active ACE2 receptors. This is the ACE2 receptors I'm talking about here on the vascular tissue. This leads to accumulation of angiotensin 2, and that leads to overactivation of the renin-angiotensin system and multi-organ injury. So that's not very helpful either. Endothelial dysfunction. We're getting a lot of studies now showing that COVID-19, particularly severe COVID-19, is an endothelial disease and can cause systemic endotheliitis. What does that mean? Well, basically vascular endothelium injury and dysfunction. And that leads on to, here we've got the macro and microvascular dysfunction and injury contributes to long COVID, important study there. And we've also got related problems of hypercoagulation. So endothelial injury and endotheliitis can trigger excessive thrombin production, and this leads to microthrombi deposition. Right, let's look at the indirect effects of SARS-CoV-2 now. And the first one of these is autoimmune conditions through molecular mimicry. So the autoimmune registry has now included long COVID on its list of autoimmune conditions. So that's quite interesting. And we saw in my earlier lecture the vast range actually of autoimmune conditions which can be caused in long COVID. And here's recent studies suggest that SARS-CoV-2 specific antibodies and autoreactive T cells cross-react with host proteins, which is exactly how an autoimmune condition starts. Mitochondrial dysfunction, we've had quite a lot on this already, or very well explained. So all I need to say is that it's impaired in COVID-19 patients. Patients can't produce their energy by the mitochondrial pathways. So if you remember from Mitochondria Day, those of you who were there, we talked about how glycolysis can be a compensatory mechanism when energy is needed in an emergency and when the mitochondria are not functioning as they should. And that's exactly what's happening here in long COVID. PET scans, just to confirm the point, have shown brain hypometabolism, which is a very good indication. Reactivation of other dormant viruses, a couple of people have mentioned these. Here's, in fact, I think this, Gillian, you mentioned this one specifically. 67% of long COVID patients were positive for Epstein-Barr, that's reactivation of Epstein-Barr, and the researchers suggested that many long COVID symptoms may not be a direct result of SARS-CoV-2, but the result of COVID-19 induced Epstein-Barr reactivation. Right, and microbiome alteration. Long COVID patients, the microbiota status was not restored to normal levels 6 months after COVID-19, but that's of course because they haven't got the benefit of seeing you or being part of Professor Thomas's trial. Here's the microbiome profile, which has become reasonably well known now in long COVID patients, all of which you could do something about. Demyelination of the brain and nervous system, yes. We know already this is a cause of multiple sclerosis, which is also an autoimmune condition, but it can also cause other neurological conditions as well. Here's a systematic review of 60 studies, which found that COVID was associated with central nervous system demyelination. Contributory factors, well, genetics, yes. I'm not going to say any more about genetics, but you've got Life Code GX sitting outside. Do go and talk to them about it. Diet and malnutrition, yes. Believe it or not, a meta-analysis showed that long COVID patients had a high risk of malnutrition. That's hospital food, probably to some extent. But also bear in mind that a long COVID patient who does not have a carer is not likely to be cooking themselves nutritious meals. Lack of exercise, obviously. Physical deconditioning while immobile in hospital, of course. And poor methylation has come up again. Here we are. Researchers have noticed an association between pernicious anemia and long COVID. And here's a study showing that SARS-CoV-2 induces increased demand for methyl groups, but simultaneously impairing their supply. So there we go. We had a question earlier about folic acid, didn't we? Right. So that's a quick run through of what I think are the most likely mechanisms of long COVID. And bear in mind that several of these can be operating at any one time. So then my own research took me in this direction, viral reservoirs, which I think is absolutely riveting. And I'd really not looked into this before. So thank you, Jenny. Bye-bye. So let's have a recap on what I've already said on viral reservoirs. So they hide virus-infected cells from the immune system. There are SARS-CoV-2 reservoirs in long COVID patients in multiple organs. When the immune system is dysfunctional or distracted, the infection can be reactivated from viral reservoirs. And there are several reports of relapse or reactivation of SARS-CoV-2 in recovered patients. Do you remember I said in my first lecture that long COVID is a relapsing and remitting condition? I suspect this may be one reason why. So are long COVID sufferers still PCR positive? Well, very often not. But that's because when you take a PCR test, or lateral flow for that matter, you're testing the upper respiratory system. Now, if the viral reservoir is there, you might pick it up. But if the viral reservoir is not there, then you won't. And the viral reservoir is most likely in the gut. And so here we are. Evidence for persistent infection is shown by prolonged viral shedding in feces. Reactivation then. Here's a meta-analysis estimated that around 12% of recovered patients tested positive for SARS-CoV-2 reactivation. So this is not 12% of long COVID patients. This is 12% of all recovered patients. And bearing in mind that this is a meta-analysis, so we don't know the time frame involved. It's probably going to be a range, something like 3 weeks to 12 weeks, something like that. It's not a million miles away from the estimate of people with long COVID. So a number of studies have shown that reactivation of dormant viruses may contribute to severe COVID-19. And reactivation of SARS-CoV-2, or indeed any other virus, can only come about when the immune system is dysfunctional or suppressed. So this can occur either through poor maintenance, so inadequate levels of vitamin D, for example, but many other micronutrients, or through the induction of CARs. Do you remember we talked about CARs earlier, compensatory anti-inflammatory response syndrome? And do you remember I said there had to be a balance between SIRS and CARs? Sorry, I'm talking about syndromes again. I really shouldn't be. Promised I wouldn't. Never mind. But if you get that balance wrong and the immune system is suppressed by too much CARs relative to SIRS, then you've got a problem. And then, of course, there is immune exhaustion as well. So let's look some more about these viral reservoirs. They are coated in biofilm. So we've heard about biofilm before with bacteria. And indeed, it was fairly recently only, 2010, that's not so long ago, that it was discovered that viruses can also build biofilm. And it does exactly the same as it does for bacteria. It hides it from the immune system. Now, I don't know whether this is precisely the same biofilm as is produced for bacteria, but the viral one, at least as found in human T-cell leukemia virus, is carbohydrate-rich cocoon structure comprising DNA, protein, and polysaccharides. And by the way, you can also get toxic metals in there because they're quite useful for binding. More on that in a minute. So here's a study which showed that by removing the viral biofilm from the surface of the infected cell, there was an 80% reduction in reactivation of the virus. So that's quite interesting. This is not COVID, though. This is the human leukemia virus, but still. So biofilm removal. So it probably tempting to try and remove the viral biofilm just to get rid of all the dormant viruses. But don't do it unless you've got a patient with an immune system which is really well supported, because otherwise you're liable to do more harm than good. But if you do want to remove the biofilm, then this looks like quite a good protocol. I myself have used Claire Labs Interphase Plus and Kirkman's biofilm defense with EDTA to get rid of the toxic metals as well, and N-acetylcysteine. Cinnamon bark oil seems to be reasonably well supported, as does lactoferrin, or indeed colostrum. Right, so let's look at viral reservoirs in other viruses. Most of the research has been done on HIV, as you might expect. So here we've got viral reservoirs containing at least 98% of all HIV infected cells, with up to 95% of them being found in the gut. So the gut seems to be a good viral reservoir for practically everything. But other viruses can also persist in the body. Hepatitis B, Ebola, Zika, Epstein-Barr, other herpes viruses, varicella zoster. Now, there's a very discrete list there, and I'm going to come back to this in a minute, because they all have what I have termed retrovirus associations without being, other than HIV, retroviruses themselves. So we'll see what those associations are in just a minute. But first of all, I want to talk about retroviruses and reverse transcriptase. So retroviruses, as we know, incorporate themselves into the human genome by employment of reverse transcriptase, which is an enzyme they carry with them for this very purpose. And reverse transcription is simply making double-stranded DNA from the single-stranded RNA viral template. So as you will remember from Mitochondria Day, I'm not very good at PowerPoint diagrams, but hopefully this will make sense. So you've got retroviral single-stranded RNA, reverse transcriptase plus other enzymes makes retroviral double-stranded DNA, which is then inserted in the human genome. Now, it may come as a bit of a shock to discover that we have retroviral elements already in our genome. Some of them have been there for generations. And although potentially pathogenic, they are rarely activated, is the good news. So these are known as human endogenous retroviral elements, or HERVs, already present. So in COVID-19, HERV reactivation and the expression of HERV proteins can contribute to perpetuating pathogenic pathways underlying severe COVID-19 and presumably long COVID, although the study didn't look at long COVID, and HERVs were also found expressed in COVID-19 post-mortem tissue. So that's all quite interesting. But the HERVs, as with any viral reservoir, can be repressed by a robust innate immune system and methylation. There we are, it's come up again. Right, so I'm going to look at selected features of retroviruses. Retroviruses have many other features other than this. So we know they have reverse transcriptase and can insert themselves into the human genome. Other viruses can predispose sufferers to develop retroviral infections. Retroviruses are immunosuppressive. They can disable the cellular protein tetherin. If you're not clear what tetherin does, I've never heard of it before, don't worry, coming onto it in a minute. HIV antibodies can help neutralize other viruses, and antiretroviral drugs can suppress non-retroviruses. Let's see how that works with our few selected viruses, which also form up in viral reservoirs. So here's hepatitis B. It's not a retrovirus, but it carries in it reverse transcriptase. So that can be in the human genome as we speak. Infection with herpes simplex can predispose sufferers to HIV. The envelope protein, glycoprotein of Ebola, contains an immunosuppressive-like domain, similar to those found in oncogenic retroviruses. Antiretroviral drugs reduce the suffering from herpes simplex and Zika virus. So that's quite interesting. I had no idea about that. Ebola and Zika can both disable tetherin. Again, don't worry, I'm coming on to that. Monoclonal anti-Ebola antibodies offer cross-protection against HIV. And so these viruses, you can see why these viruses, I've said they all have retroviral associations. So let's look and see how these retroviral associations work with SARS-CoV-2. First of all, can it be reverse transcribed? Yes, it can. So in this study, which is, wait a minute, there we are, by Zhang et al., they found that it could be reverse transcribed and integrated into infected human DNA. The researchers expressed concern that SARS-CoV-2 might haunt our cells long after the infection is gone, which is quite a good description of long COVID, really. I think it's important to say, though, that not everybody agrees with the findings of this study. It's not on scientific grounds because there is absolutely nothing wrong with the science. Nobody can criticize the study, but it's not yet been replicated, largely because people are too terrified to replicate it because of the implications. So we may get replication of the study later, or we may not. But so far, there's one study showing that SARS-CoV-2 can be reverse transcribed into our own DNA. Can it disable tetherin? Yes. So that aids its release from the SARS-CoV-2 release from cells and enhances viral spread. Loss of tetherin from the cells caused an increase in viral titers. Is it immunosuppressive? Yes, it is. The immune system failed to launch robust interferon responses, and insufficient interferon resulted in higher viral loads in host cells. And here's the antibodies. Non-neutralizing antibodies could enhance SARS-CoV-2 infection through antibody-dependent enhancement. Neutralizing HIV antibodies here can bind to the glycosylated spike protein to help neutralize it. So that's HIV antibodies helping out in SARS-CoV-2. So let's see where that leaves us. SARS-CoV-2 is found in viral reservoirs, particularly the gut. It can be reverse transcribed and may already be in the human genome. It's immunosuppressive, reducing the interferon response, increasing viral load, and increasing the level of non-neutralizing antibodies, in other words, the ones that make things worse. It can disable the interferon-induced protein tetherin, whose expression blocks the release of envelope viral particles. Disabling tetherin enhances viral spread. SARS-CoV-2 can have retrovirus associations, as with the ones that I went through earlier. And interestingly, this tends to support Luc Montagnier's paper, showing that RNA fragments of retroviruses were found in the SARS-CoV-2 genome. And that brings me back to a study which I showed this morning. Thaler et al. Do you remember this one? 3 to 4 months after symptom onset, the predictor of long COVID versus non-long COVID was a stronger interferon response. So if you're not clear on what interferon does with respect to viruses, you can read about it there. What is the relevance of interferon in COVID-19? So during severe infection, there is robust production of pro-inflammatory cytokines and chemokines with limited production of interferon. Minimal amounts of interferons have been detected in the peripheral blood or lungs of patients with severe COVID-19. So that seems to bear out the hypothesis. Large study here, the COVID-19 pneumonia patients, so definitely severe, they had neutralizing antibodies against interferon. It's the first time we've heard about that, but that's not going to be good news for sure. These inhibit the ability of interferons to block the SARS-CoV-2 infection. And SARS-CoV-2 encodes several proteins that specifically invade interferon type 1 response. So interferon, of course, can be given as a pharmaceutical treatment, but it's got side effects, fatigue and headaches being the main ones. Happily, we can do it naturally with all of these. And if you're wondering if there is something missing from this list that really should be in there, Damien, there is. It's vitamin D, of course. There's a lot of studies showing vitamin D enhances the interferon response, particularly with magnesium and zinc. Magnesium, of course, activates vitamin D. Zinc, again, is vital for the immune system. So, what can we conclude from all this? So there are many potential mechanisms underlying long COVID, and we can have several active at any one time, I would suggest. And there may be many more that I have failed to mention. A likely but relatively unexplored candidate is the reactivation of viruses. And of course, this can be SARS-CoV-2, but also any other virus that happens to be in a viral reservoir, normally the gut. This explanation allows for the initiation and or continuation of viral symptoms, as well as the relapsing and remitting nature of long COVID. It allows for patients to be PCR negative in the case of SARS-CoV-2 reactivation, unless the viral reservoir happens to be in the upper respiratory tract, which it usually isn't. So in common with some other retrovirus associated viruses, which we went through, SARS-CoV-2 can probably insert itself into the human genome via reverse transcriptase. It is immunosuppressive. It disables tethering, all of which enable viral spread. Immunosuppression and the disabling of tethering prevent an appropriate interferon response, and this may be the determining factor in whether a patient develops long COVID or not. Various natural substances, particularly vitamin D, can actually boost interferon production. And that's it. Thank you very much.